Hello, dear colleagues. I want to make a report on the topic of genetics aspects of the conservation of honeybee populations of Apis mellifera mellifera and its microbiome features. My name is Rustam Ilyasov. I am from Moscow, Vavilov Institute of General Genetics. Honeybees are the most active pollinators on the planet. They pollinate about 80% of flowering plants and about 60% of agricultural crops. Honeybees belong to family of Apidae. The Apidae family belongs to the order Hymenoptera and includes about 5,000 species. The most of famous members of the family are the honeybee, Apis mellifera, the Asian bee, Apis cerana, and bumblebee, Bombus terrestris. The honeybee colonies consist of 100,000 workers, 500 drones, and one queen. The honeybee Apis mellifera species originated 1.5 million years ago in Southeast Asia. Subspecies appeared 10,000 years ago. The species is subdivided into 30 subspecies. There are 11 subspecies in Europe. In Africa, 12 subspecies. In the Middle East, there are 7 subspecies. All subspecies subdivided into 6 lineages. Almost all Africa inhabited A lineage, M North and Western Europe, C Southern and Eastern Europe, O and Z Middle East, Y Northeast Africa. Russia and West and North European countries naturally was inhabited with one common honeybee subspecies, Apis mellifera mellifera, dark European bees. Now this subspecies is subject to intensive hybridization with subspecies from southern and eastern Europe. Only a few isolated populations of dark forest bee have survived. In the Ural and Volga region remain the largest populations, more than 600,000 colonies of the dark forest bees. In the Republic of Bashkortostan, the Burzen population has remained in the mountain forest area, which is kept in the old style in the hollow of trees. Dark forest bees highly adapted to long forestry winters and can survive in hives over 6 months without flights. In order to conservation of honeybee subspecies, identification is important. Morphometric and allozyme polymorphism, the first markers in population studies of honeybees. These markers are not useful in the identification while hybridization. Further, molecular genetics methods are developed. These methods are based on mitochondrial and nuclear genomes. Genetic markers are allelic polymorphism, restriction polymorphism, nucleotide sequences polymorphism, and single nucleotide polymorphism (SNP). The dark forest bees can be successfully identified using these molecular genetic methods. The nuclear genome of honeybee Apis mellifera is 246 million base pairs, subdivided into 16 chromosomes and contain 10,000 genes. The mitochondrial genome of honeybees Apis mellifera is about 16,000 base pairs, is represented by a circular DNA molecule and contain 35 genes. It is map of the Ural and Volga region. Ufa is capital of the Republic of Bashkortostan. Red point is a geographical localization of the sampling places of the bees in the Ural and the Volga region. The Republic Chuvashia, the Kirovskaya Oblast, the Perm Krai, the Republic of Udmurtia, and the Republic of Bashkortostan. In the Republic of Bashkortostan, we collected honeybees from 2,309 colonies in 398 apiaries. We collected bees from 24 districts in the Republic of Bashkortostan. In the Perm Krai, we collected honeybees from 362 colonies in 41 apiaries. In Sverdlovskaya Oblast, from 58 colonies. In the Republic of Udmurtia, from 200 colonies. In the Republic of Tatarstan, from 52 colonies. In the Kirovskaya Oblast, from 64 colonies. In the Republic of Chuvashia, from 14 colonies. Caucasian region used for collection of bees, southern subspecies A.M. caucasia and A.M. carpatica. In total, we collected honeybees from 3,123 colonies from 493 apiaries. Study the differentiation of mitotypes of bees of the evolutionary branch M on the basis of the analysis of polymorphism of the nucleotide sequence of the CO1, CO2 locus of mtDNA. The nucleotide sequence of the mtDNA CO1, CO2 locus was analyzed in 109 samples of bees, of which 20 colonies from 18 apiaries in 6 districts of the Republic of Bashkortostan and Perm Krai. We constructed the median network of genetic relationships of bees of the evolutionary lineage M based on the SNP analysis of the mtDNA CO1, CO2 locus. The samples of AM mellifera from the Ural have a red color and located separately from European samples of AM mellifera. Most of European of AM mellifera mitotypes belonged to the central group. One European mitotype M10 located together with samples from the Ural region. Classification of M mitotype of honeybees based on the analysis of SNPs of CO1, CO2 locus of the mtDNA. We subdivided all M mitotypes into 8 groups. The samples of AM mellifera from Ural combined into group 2 and differ from European by SNP 912 GA. The phylogenetic study of Apis mellifera subspecies based on the analysis of polymorphism of sequences of the gene ND2 of mtDNA and analysis was carried out on 117 bees of 20 subspecies of 4 evolutionary branches. An analysis of the genetic relationship of bees from the 4 evolutionary branches AMCO was carried out on the basis of a joint analysis of 12 genes of mtDNA. The nucleotide sequences of the mtDNA ND2 gene of 39 samples were determined by us and 78 samples were taken from the gene bank. We constructed the dendrogram of genetic relationship of honeybees from evolutionary branches based on the genome-wide comparison of complete mtDNA. The samples of honeybees from different evolutionary branches located separately. In the table presented single nucleotide polymorphism SNPs in 12 genes of mtDNA between honeybees from 4 lineages. C lineage was used as a reference. Highlighted genes are most informative for honeybee subspecies differentiation. The dendrogram of genetic relationship of subspecies of bees was constructed based on the analysis of the nucleotide sequence of ND2 gene of mtDNA. All samples were subdivided into 4 groups which matched to lineages AMCO. The lineage C divided into 2 groups according to their origin. The median network of genetic relationship of honeybee subspecies was constructed based on the analysis of SNPs on the ND2 gene of mtDNA. Here, all samples are subdivided into 4 groups: AMCO lineage M, located between A and C, and differs from them by SNP 634 TC and 816 CT, respectively. Thus, ND2 is informative markers for honeybee subspecies differentiation. Phylogenetic study of Apis mellifera subspecies based on the analysis of polymorphism of nucleotide sequences of the gene vitellogenin VG of nDNA. Here, the analysis performed on 12 samples of honeybee of subspecies Apis mellifera on the oral populations of the evolutionary lineage M, and nucleotide sequences of 6 exons of the gene vitellogenin VG were obtained. Comparative analysis was performed on the 28 samples of honeybees from 4 subspecies retrieved from the gene bank. We detected following single nucleotide polymorphisms SNPs in 6 exons of the VG gene and AM mellifera from the oral in comparison with sequences of AM ligustica in gene bank. Highlighted 16 SNPs are non-synonymous. Based on the comparative analysis of gene VG exon sequences on 28 samples of honeybees from 4 subspecies retrieved from the gene bank and our sequences of VG exons, we constructed a phylogenetic tree. Here, all samples subdivided according to their belonging to the evolutionary lineages. Thus, VG are informative marker for honeybee subspecies and AM mellifera identification. We compared gene VG exon sequences of AM mellifera and AM ligustica and detected following unique SNPs in each exon. These SNPs are markers for identification AM mellifera, highlighted duplicated SNPs. Thus, gene VG can be used for identification dark European bees. The assessment of introgression of southern genes and genetic characteristics in the population of dark forest bees in Ural and Volga region. The polymorphism of 9 microsatellite loci of nDNA as well as CO1, CO2 locus of mtDNA was studied in 3,123 honeybee colonies from 49 regions in the Ural and Volga region and 3 southern region of North Caucasus and Eastern Carpathians. We assessed the introgression level in the local honeybee populations based on the polymorphism of 9 microsatellite loci of nDNA loci and CO1, CO2 loci of mtDNA, locus of mtDNA. Yellow is C lineage genes. Blue is M lineage genes. Here presented introgression on nuclear and mitochondrial genome levels. On the table presented the values of heterozygosity and inbreeding coefficient in first 24 populations of honeybees in the Ural and Volga region based on the polymorphism of 9 microsatellite loci. On the table presented the value of heterozygosity and inbreeding coefficient in following populations of honeybees in the Ural and Volga regions based on the polymorphisms of 9 microsatellite loci. All purebred populations of AM mellifera are characterized with little deficit of heterozygosity and inbreeding. All hybrid populations are characterized with little excess of heterozygosity and inbreeding. We constructed phylogenetic tree based on the polymorphism of 9 microsatellite loci of nDNA in order to show the differentiation of local honeybee populations. All populations subdivided into 4 groups. Green color defined purebred populations, red populations of southern subspecies, yellow populations with little level of hybridization. Those microsatellite markers are able to differentiate purebred and hybridized populations. In the box presented the standard genetic characteristics of the purebred dark forest bee A.M. mellifera. All purebred populations of A.M. mellifera are characterized with little deficit of heterozygosity and inbreeding. On the geographical map, we presented the distribution of southern genes in the population based on the analysis of polymorphism of 9 microsatellite nDNA loci and the mtDNA CO1 CO2 locus, each circle divided into 4 sections of M and C lineage gene fractions in nuclear and mitochondrial genomes. White and black is M lineage, and yellow and blue is C lineage, and both genomes respectively. In the Ural and Volga region, we defined 5 remaining populations of A.M. mellifera: 1 Burzянская, 2 Tataslinskaya, 3 Южно-Прикамская, 4 Vysherskaya, 5 Kambarskaya populations. The assessment of introgression level of southern genes and main genetic characteristics in the colonies of the dark forest bees in Ural and Volga region. The polymorphism of 9 microsatellite loci of nuclear DNA and the mtDNA CO1 CO2 locus was analyzed in 25 colonies of dark forest bees A.M. mellifera from the Burzyan district of the Republic of Bashkortostan. We assessed the fractions of genes of lineages M and C in colonies of Burzyan bees based on the analysis of polymorphism of the CO1 CO2 locus of mtDNA and 9 microsatellite loci of nDNA. The hybridization on nuclear and mitochondrial genome levels is shown separately. Most of colonies in Burzyansky district in is purebred, but 2 highlighted colonies are hybrid with southern subspecies. We presented on the plot fractions of genes of lineages M and C in colonies of Burzyan bees based on the analysis of polymorphisms of the CO1 CO2 locus of mtDNA and 9 microsatellite loci of nDNA. The hybridization of nuclear and mitochondrial genome levels is shown separately. Most of colonies in Burzyansky district is purebred, but 2 colonies in the red circle are hybrid with southern subspecies. We assessed genetic characteristics in colonies of Burzyan bees based on the analysis of polymorphism of the CO1 CO2 locus of mtDNA and 9 microsatellite loci of nDNA. The genetic characteristics of hybrid colonies are yellow highlighted, which differ from purebred colonies of A.M. mellifera. The purebred colonies of A.M. mellifera are characterized with the little deficit of heterozygosity and inbreeding. The hybrid honeybee colonies are characterized with excess of heterozygosity and outbreeding. We constructed the phylogenetic tree for Burzyan bees colonies based on the analysis of polymorphism of the CO1 CO2 locus of mtDNA and 9 microsatellite loci of nDNA. Red color defined hybrid colonies, green highly purebred colonies, yellow purebred colonies with little integration of southern subspecies genes. Thus, we, based on microsatellite loci, recognized purebred colonies and colonies with different level of hybridization. Based on the analysis of polymorphisms of the CO1 CO2 locus of mtDNA and 9 microsatellite loci of nDNA, recognized purebred colonies and colonies with different level of hybridization. We divided colonies into 3 groups: red color defined hybrid colonies, green highly purebred colonies, yellow purebred colonies with little integration. Colonies from yellow and red groups shouldn't be selected for further breeding. We are suggesting a method of selection of dark forest bee colonies with integration of southern genes, at most 5%, using the genetic characteristics of purebred colonies of AM mellifera, heterozygosity, and F statistics. We studied the microbiome of dark forest bees of Ural and Volga region, the gut microbiome of bees from 25 colonies of the dark forest bee subspecies AM mellifera from the Bursan district of the Republic of Bashkortostan was analyzed. Also, we provide analysis of antimicrobial activity and quality of the light and dark honeys. The gut microbiome of honeybees Apis mellifera located predominantly in rectum. The content of microbiome is simple and contains about 8 species: Lactobacillus ferm 5, Lactobacillus ferm 4, Villamilla apicola, Strongylophila alvei, Bifidobacterium spp, Trishula pereira, Parasaccharobacter apium, and Bartonella apis. Our aim is isolation and identification of the bacteria on the genus Lactobacillus from the honeybee Apis mellifera for creation of a probiotic preparation that will increase the resistance of honeybee colonies to pathogens. Probiotic microorganisms protect honeybees from pathogen microorganisms using organic acids, ethanol, benzoate, enzymes, hydrogen peroxide, antimicrobial peptides, bacteriocins. Honeybees provide bacteria with niche and food. Honeybee and microbiome help each other. Lactobacillus are symbionts of honeybees, Apis mellifera. Lactobacillus are immobile, catalase-negative, non-spore-forming, rod-shaped polymorphic representatives of the order Lactobacillales, family Lactobacillaceae. In the gut of honeybee detected following lactobacteria: Lactobacillus kunkii, FON2, Lactobacillus apinorum, FOM13, Lactobacillus mellis, HON2, Lactobacillus melliferpin 4, L. cula-bergensis, BIOT2, L. kimbladii, HMA2, L. helsinga-bergensis, BMA5, L. milliventris, HMA8, L. apis, Lactobacillus apis, HMA11, Butler et al., 2014. Here presented showing the contents of the intestines of the honeybee for the detection of Lactobacillus. We used sector seeding methods, cold method. On the plate is shown honeybee gut microbiome growing. We cultivate the contents of the gut of the honeybees. We used agar AGV to detect the diversity of microorganisms in the content of the intestine of a honeybee. The gut contents of the honeybees were put on lactobac agar medium for detection and cultivation of Lactobacillus bacteria. The results of bacteriological inoculation of the gut contents of the honeybee are presented on the table. Here is shown a growth dynamic of microorganisms from the honeybee gut on HGV agar in 1, 2, and 3 days. Honey used for comparison. Also here is shown the results of showing the contents of the honeybee gut for the detection of Lactobacillus on the lactobac agar medium. On the other table presented the dynamics of growth of Lactobacillus from the gut of honeybees on the lactobac agar medium in 1, 2, and 3 days. There are 3 stages of sowing: 1. Intestinal content sowing, subseeding of the obtained biomass; 3. Sowing pure strains. In order to detection of Lactobacillus of the gut of the honeybees, we isolate colonies of putative lactobacilli marked with numbers 1 to 7 from plates to tubes. We provided determination of biochemical activity of isolated from the honeybee gut puer cultures of Lactobacillus. All isolated puer cultures were microscopic examined, which stained according to gram positive. All bacterial colonies was gram positive. Here we present the determination of carbohydrate fermentation of 7 isolated bacterial colonies after 18 and 48 hours of incubation. The determination of carbohydrate fermentation was carried out using carbohydrate discs and broth with bromcresol pepple. Catalase activity of puer cultures according to GOST 30,425. The result is negative in all species since the indicators are below 0.1 thousandths. These bacteria belong to Lactobacillus and be used as probiotics in beekeeping. Further, we analyze the antibacterial, antimicrobial activity and quality of honeys. Here presented the analysis of the honey properties in our laboratory. The botanical origin, pollen analysis by microscopy and PCR. Physiochemical properties, analysis of the pollen spectrum, analysis of the protein spectrum of honeys, comparisons of fresh honey, unripe honey, and 2-year-old honey. Antibacterial activity of honey by growth. Suppression zones of E. coli, probiotic properties of honey in comparison with glucose. On the tables presented analysis of antibacterial activity by inhibition the diameter in millimeters of the honey against E. coli XL blue, Bacillus subtilis, and E. coli GM109. Different kinds of honeys were analyzed. 1 floral honey, 2 dark honey, delight honey, cell honey, linden honey, buckwheat honey. The linden honey, buckwheat honey, and cell honey shows highest antibacterial activity. Here presented comparison of enzyme activity of dark buckwheat, chestnut, thyme, and light linden, melilotus, polyfere honeys. Dark honeys have higher enzyme activity than light honey. The high content of enzyme and proline are indicator of the high quality of honey. We did the microscopy analysis of honeys of the Republic of Bashkortostan. The pollen contents of the 3 honeys with 78, 65, and 74% content of linden tree pollens are presented on microscopy. Thus, in Bashkortostan, dark forest bee produce unique high-quality linden honey. In the Republic of Bashkortostan, 10 years ago, established an autonomous non-commercial organization, Altınsuluk. The project Altınsuluk, based on the scientific data, released purebred beekeeping with dark forest bees in the Republic of Bashkortostan. The director of Altınsuluk is Marat Hasanov. Altınsuluk is inter-regional center for the protection and promotion of beekeeping products of the Republic of Bashkortostan. In Russia, the livestock of Altınsuluk is 1,000 dark forest bee Apis mellifera mellifera colonies. The number of honeybee colonies which they protect participants is more than 4,000. The number of honeybee colonies with the all private beekeepers is more than 15,000. The total number of beekeepers involved in the project is 60, of which 10 beekeepers are working on purebred honeybee reproduction. Currently, Altınsuluk has 5 purebred isolated apiaries on dark forest bee Apis mellifera mellifera in 5 districts of the Republic of Bashkortostan in Russia, Ural. It is shown on the map of the Republic of Bashkortostan. Altensolog provides a careful check the genetics and microbiome of our honeybee colonies in the laboratory, careful check the biochemistry, microbiology, and pollen composition of our honey in the laboratory, careful selection of honeybee colonies for economically useful traits in isolated purebred apiaries. Ministry of Agriculture able to provide certificates and accompanying documents for honey and honeybee colonies from Altensolog. Altensolog is able to the export of high-quality honeys and purebred queens and colonies of A.M. mellifera to countries where there is a demand. Thank you very much. Your questions. Goodbye.